Dr. Kirill Lobachev got his Ph.D. in biology from St. Petersburg State University in Russia. He's been here about 6 years, I guess.
Yes.
Now I'm associate professor in biology. Some of you may have noticed the blackboard outside IBD. Kirill Lobachev says we have the best sandwiches on campus. He's a part-time food critic for the cafe. I don't know if he gets his free food there or what the payment is.
Not anymore.
Anyway, it's a real pleasure to have Kirill here, and I look forward to learning about his research. Turn on the lights for you.
Thank you very much for inviting me. It's a great pleasure to make presentation in this building. I think it's a wonderful set of events, and ironically, for the time I've been here for 6 years, it's my first time when I have the pleasure to make a presentation in this audience, in this auditorium. My title of my talk, you can see, is pretty long, and I can end essentially here because it summarizes everything I'm going to talk about. So you can see that we are focused on studies of chromosome stability, and we carry out our research in yeast. At the same time, we think that these studies are important because we can extrapolate these results to problems that people are dealing with, studying human diseases and human polymorphisms. This building, as you know, it contains a lot of labs that are doing different research, and recently I've been invited to be on Ph.D. committees for several people who are doing absolutely different things, such as studying bone formation, bone restoration, and obviously the studies are very far away from what I'm doing if you look at this title. At the same time, after being in these committees for a while, I understood that there are some similarities, and even at some approximation, we're using similar terms and studying similar problems. So if you imagine that if you come to the statement that chromosomes are bones, and depending how you draw them, they even look alike. So I realized that people who are studying how bones are broken and how they're repaired, they encounter the same questions that we're trying to address when we study chromosome breakage. First task is you need to repair these breaks in both cases, and these breaks must be repaired pretty accurately. Otherwise, if you don't repair the bones, that can lead to lethality, or if you repair it inaccurately, it can lead to a person with disabilities. The same applies to chromosomal breakage. So there, in some sense, it's a scaffold from genetic term, because they carry a lot of genes, essential genetic information. So if break is not repaired, it leads to cell lethality, cell death, or if it's repaired incorrectly, it leads also to disabilities that are manifested by hereditary diseases and cancer in humans. The questions that are addressed, and that's questions I encountered by being on these committees, are almost the same what people are asking when they study chromosomal breaks. Why bones or chromosomes break? What genetic and environmental factors predispose them for breakage? How to repair these breaks efficiently, and what goes wrong when repair is inaccurate. This is so-called spectral karyotyping of human cells, normal cells. Each cell is depicted by a specific color. Those are very precious carriers of our genetic information, and there are a lot of mechanisms in cells exist that guarantee that these chromosomes are going to be stable and intact. But sometimes something goes wrong, and if you look at some cancer cell lines, you can find that chromosomes diffuse to each other. So if you see two colors next to each other, it means it's a rearrangement. Most probably it's translocation. As a matter of fact, majority of cancer cell lines, they're characterized by structural or copy number variations in why it's all happening? And probably among all the damages that can drive chromosomal instability, double-strand break is the most dangerous type of damage because one unrepaired break can drastically change the level of expression of genes. For example, if the broken fragment will be not repaired and lost, you can lose the wild type allele that covers mutant allele on another chromosome, the process that is called loss of heterozygosity that can lead development of cancer. At the same time, unrepaired break can be under selection. It can be amplified because it doesn't contain centromere, and now it can, by stochastic process, it can accumulate in the cells, and that leads to gene amplification. Example here is c-MYC, oncogene transcription factor, that drives the cancer development. And very often, this extra chromosomally amplified regions, they look as inverted dimers. They call double minuets. Another consequence of the break that this broken fragment, it becomes very prone for invasion, so it can invade non-homologous chromosome, and it can lead to translocation events. And one of the most famous examples is the formation of BCR-ABL fusion kinase that is expressed in chronic myelogenic leukemias. And the only successful drug on the market right now that partly treats this leukemia development is Gleivec and TIP exist. At the same time, this double-strand breaks can lead to smaller scale variations, and that became very popular recently. Apparently, the human population is loaded with so-called copy number variations. It means that certain genes that either, if you look at different individuals, we can have more of some genes or less of some genes. Even twins are different in so-called copy number variations. And these copy number variations, of course, they contribute to diseases, but at the same time they contribute majorly to polymorphisms, and why we look different from each other. So what agents can break chromosomes? What agents can break phosphodiester bonds on DNA level? There are a lot of them. They're very well characterized, and there are a lot of exogenous factors or endogenous factors that can break DNA directly or indirectly, for example, as ionizing radiation. And assumption is, and for a long time, people believe that this is probably the most prominent source of DNA damage. And these agents that can break DNA at any given position, essentially any region can be prone for this breakage, except for the case of good breaks that are introduced by site-specific endonucleases. But other agents, they are more or less, they lack specificity. Nevertheless, in maybe last decade, it became clear that breaks don't happen randomly, and certain regions, they're more prone than others. And people see it on evolutionary scale, or they can see it when they analyze chromosomes from patients that suffer from cancer or from hereditary diseases. If you look closely, when you look at the regions that are predisposed for breaks, you can find that very often they contain repetitive elements. And if you analyze growth rearrangement breakpoint database or human gene mutation database, those are repositories of all information when from sperm or somatic cells carrying translocation events. Very often you can find repetitive elements or DNA repeats. But not only simple repeats, but rather a lot of cases you find repeats that can adopt so-called non-beta DNA conformation. I'm going to talk about it later. More specific examples is Fragile X syndrome that is discovered by Steve Warren. So that results from expansion of CDG repeats, and this is metaphase chromosome, X chromosome, and you can see that teeny tiny links here that very often break under cover slip. They actually break and dissociate in vivo in live cells because it leads to so-called Jacobson syndrome. So these males are characterized by very severe deficiencies. The frequency of carriers that carry so-called permutation size repeats, that's 60 repeats. 1 out of 200 women in the human population, they carry this so-called permutation size repeats. I think something is not working anymore. I'm try to speak up. And that's not the only place in genome, not only in chromosome X, but other positions across genome where expansion of the CD-rich sequences can happen and predisposes the regions to fragility. Another example is translocation between chromosome 14 and 18, and that is very common translocation in follicular lymphomas, and it's caused by breakage at BCL2 region, which contains triplex forming repeats. And last, but definitely not the least example is constitutional recurrent translocation stimulated by breakage at palindromic sites. That translocation between chromosome 11 and 22, when two palindromes are joined to each other, and usually it doesn't cause disease, but it leads to human infertility. So far, there are 7 AT-rich palindromes identified in human genome. They are scattered around the genome. However, it's known that it's very difficult to sequence them, and how many of these unstable sequences exist in the human genome is still a big mystery. What is conclusion from these cases is that these repeats can occur de novo, or we can inherit them from our parents. And then if you look what is common feature of the sequences that are implicated in causing breakage, in causing rearrangements, the common feature is that all of these repeats, they can adopt so-called non-canonical DNA structures, such as hairpin structures or cruciform structures, triplex or quadriplex DNA. Those are different conformations that they differ from canonical duplex. How this breaks, how the structures lead to breakage is still a mysterious process. And if you modify these questions that were addressed to study bone damage, chromosome damage, we added a couple more. So in our studies, we want to understand how potent are this secondary structure adopting repeats and inducing chromosomal breaks. It means that we can find it in 1 out of a million cells. Every cell is going to experience this break if the cell contains this repetitive sequence. Why chromosomes break? So why architectural change in DNA organization can cause such drastic consequences? Why chromosomes break? What factors mediate it? And obviously, what structural parameters of repeats make them prone for chromosomal breakage? Not all repeats that you're going to look like, you're going to analyze, are going to cause double-strand breaks. These questions we're trying to address in our work, in our studies, and in my lab we're studying XV sequence motifs formed by palindromes. So this is cruciform structure. So invert repeats, they can adopt cruciform or hairpin structures, triplet repeats, and recently, in collaboration with Katie Roth lab from Emory University, we're studying TD-rich repeats. And I decided, when I thought about how I'm going to organize this talk, I decided not to talk about all of these repeats because otherwise it's going to be overwhelming. I'm going to take an example and just to show you the way how we analyze the breakage that is formed at the cruciform adopting sequences, and also what are the methodology we're using, how we analyze the consequences of these breaks and the mechanism of this breakage. But essentially, the same approaches we're using to analyze breaks that are happening at these motifs as well. And just to be ahead of me, if you look at the mechanism of breakage, for each repeat, for each structure, the mechanism of breakage is different. And these repeats, they leave particular signature, how breaks are happening. And if you analyze the resulting rearrangements, you can play it back and deduce what was causative factor for the instability. Okay, we're using, we're not working with humans, we're using yeast as a model organism. So far, at least in the DNA repair field, it's known that whatever you discover in this model organism, it stays the same, stays true for mammalian genomes, at least in DNA repair. The way how we detect breaks, we had to build a system, and we wanted to build a system which is going to be very easy and also very, very sensitive to detect chromosomal breakage. So what you see here is the diagram of particular chromosome, chromosome 5. This circle, it means centromere. This is the left arm, not to scale, of this chromosome. And first person who noticed that there is something particular about this chromosome is Richard Kalodner, who found that there's a naturally present CAN1 gene on this chromosome. And the beauty of this location is that if you go to the left side from CAN1, it doesn't contain any essential genes, which means that if break happens in this region, yeast, they're going to survive, and CAN1 is counter-selectable marker. It's like a drug. So I'm not going to go into yeast genetics, but essentially, if we select on the media containing canavanine, we can select for events when chromosome is going to be broken and CAN1 is going to be lost, and we can monitor it using additional marker. Yeast nicely turn their color, so they usually they're white, they become red, and here's our insertion site. We can insert any sequence motifs and to analyze how frequently this chromosomal arm is going to be lost. So essentially, it's going to look something like this. Spontaneous rate of arm loss is extremely rare, is extremely low. It's 1 to 5 to 10 to the 9. So in microbiology terms means that you need at least to replica plate 20 plates, and maybe you're going to find one revertant. It's a very, very rare event. In cartoon way, we're looking for something like that. So essentially, the arm is lost, and then we're going to look how this chromosome is going to repair this broken arm. Also, we have two additional markers, CAP1, SFA1. Those are so-called gene dosage markers. Usually, yeast, they will not grow on the media containing copper. And here we have CAP1 gene, which is encodes for protein that chelates copper. But they can grow on the media with high concentration of copper if gene amplification happens. Essentially, we're going to look if this broken fragment is going to be amplified. And again, spontaneous rate of gene amplification, extrachromosomal gene amplification, is very, extremely low. So as I told you, we are studying palindromic sequences or inverted repeats, in other words. In a linguistic way, those are very funny sequences. They read the same from left to right or right to left. This is the oldest palindrome. It's written on a fountain in Hagia Sophia temple in Constantinople. So, but it's in Greek. It's going to be the same sentence from left to right and right to left. In English, if you translate it, it says, wash your sins, not just your face, before you enter this church. Since DNA has polarity, so it's going to be the same sequence that it reads from 5' to 3' direction in upper strand and from 5' to 3' direction in bottom strand. Due to this symmetrical organization, it's believed that approximately 10 base pairs in a center of symmetry must melt to allow this intermolecular interactions. So the C can pair with G, T will pair with A with T, and that is a process is called extrusion of cruciform. Normally, it doesn't happen. You need to have certain level of negative superhelicity, and obviously the composition of nucleotides at the center of symmetry matters. So more T's you have, the more easy this structure will extrude. So if it happens in one strand, it will lead to formation of hairpin. If it happens in both strands, it will lead to formation of so-called cruciform structure. We decided to look at palindromes or quasi-palindromes composed of ALU repeats. ALU repeats, they're 300 base pair long. There are a lot of them in the human genome, so we have about 1 million copies of ALU repeats, the so-called junk DNA in the human genome, which comprises 10% of the total human DNA. And very often ALU repeats, they form clusters where they're closely spaced. So this is the diagram of human HPRT gene, and these triangles here, red ones, they depict ALU elements. In some cases, they can be relative to each other, can be indirect or in inverted orientation, and they're located at a certain distance from each other. Also, they're very often they're diverged; they're not completely homologous. So here, in the LICE2 gene, which is next to CAN1, our counter-selectable marker, we inserted ALU elements separated by a very short spacer, 12 base pairs, and they differ in level of homology. They can be completely homologous, a little bit diverged. It means that these two sequences there differ from each other. And as a control, we used absolutely the same repeats, indirect orientation. So these repeats, they cannot adopt any structures. And then we assess how frequently this arm is going to be lost. And if you look at strains, analyze strains containing direct ALU repeats that can not adopt any structures, this is how it looks like. I hope you can see it. It's essentially replica plating from rich media to selective media, and we can see very few white colonies. And these white colonies indicate that mutation in CAN1 region happened, but we don't see any red colonies here, which means that in this strain, the arm loss event it happens with very, very low frequency. But if we insert inverted repeats, 100% homologous, then we can see drastic effect. It's 25,000-fold. And look at this microbiological level. Look at this colony. It's just completely covered with white papilla, with red papilla, which indicates that indeed the cells, they keep losing arm with incredibly high frequency. What's amazing about it, when I look at this, that we're not treating cells with any DNA damaging agents. So we're not treating them with ionizing radiation. We're not cutting it with any endonuclease. What we did, we just flipped one repeat relative to each other. And then, bingo, you're going to have chromosome that becomes extremely, extremely unstable. This is absolutely sequence independent. It doesn't matter what repeats you're going to create, what palindrome you're going to create, consisting of ALU repeats, or we played with other gene, URAS3 or TETO perfect palindrome, effect is the same. And also, if we decrease level of homology, breaks are still happening. You see they're happening not as frequently in case of 100% homologous repeats, but they're still happening, and they're happening with quite high rate. How these broken chromosomes look like? If you look at the arrangement of this region, simplistic way of looking at this, if this chromosome 500 kilobase long. If this region is going to be lost, and its distance between telomere to the breakage site is 43 kb, it's going to become 43 kb shorter, right? It's a very simple assumption. But if you run it on the gel and highlight these regions with the probe, this is wild-type chromosome. It's 585 kb long from left and right. But majority of these chromosomes, they run with delay, which means that they not only lost genetic information, but also gained genetic information, except for this clone. And how does it happen? To look at these chromosomes, their structure, we use so-called comparative genome hybridization on microarrays. That's a very powerful technique where you can isolate DNA from wild-type strains and from strains that lost arm, share this DNA, label it with different colors, mix it to 1-to-1 ratio, and expose it to DNA microarrays when we have all yeast genes aligned here on each spot. That's how it's going to look like at the end. So each lines here represent yeast chromosome, and each bar on the top, on the bottom, it depicts open reading frame. This is chromosome 5. We're looking yeast. They have 16 chromosomes and mitochondria right here. So yellow color, it means there is no change in copy number. Blue color, it means the region is gone. Red color, it means it's at least duplicated, only present in multiple copies. And this is one of the isolates. You can see that indeed, as we expected, this 40 kb region is gone. So it turned blue. At the same time, right next to it, there is a duplication. And there is one more duplication somewhere else on another chromosome. So if you look at what is common between these long chromosomes, if you look at chromosome 5, they almost look identical. So indeed, as we expected, based on our genetic microbiology data, they become resistant to cannabinoid. They lost adenine. It means that the arm loss is indeed happened, and indeed it happened. At the same time, right next to it, there's a duplication. If you look at this, you can use it as a signature for these breaks. So if invertebrates break, that's what they're going to look like. We know a little bit more about these breaks. We know that the way how breaks are happening at the location of invertebrates, they're happening in a very unusual way. Apparently there is nuclease, and we still don't know what is the identity of this nuclease. It doesn't depend on many enzymes that are known in DNA repair world that are nucleases or topoisomerases that potentially can target the structure. Nevertheless, none of these known players they cut the structure, but there's someone else, and there's some protein. It makes symmetrical cut, and after the cut is done, it's also maybe in conjunction with the enolagase, and maybe it has its own property. It converts these broken fragments into hairpins. So it's not a break with the broken ends. They're covalently closed, completely protected. But we know that there are nucleases that should open these hairpin cap breaks. But if they don't open, or sometimes there are too many breaks and they cannot deal with all of them, these hairpin cap molecules, they become templates for DNA replication. If one end of the linear molecule is covalently closed, so you see that bottom and upper strand, it can become template for DNA replication. So if DNA replication happens, it's going to open up the structure. It's going to lead to formation of so-called inverted dimer. That's the full mechanism how it's happening. So invertebrates, they form cruciform. It's cleaved symmetrically. Then you form hairpin breaks. Then they can be replicated to form dimer. On a chromosomal level, since this is telomeres, this is centromere, this break is going to lead to formation of acentric fragment and centromeric fragment. Since we're following the loss of acentric fragment, we're going to focus on this centromere containing fragment. If this hairpin is going to escape attack from this nucleases, it's going to lead to formation of dicentric. And dicentric is going to break. And break is going to happen not right in the middle. It can, because it's mechanical break. This is break formed by forces that pull centromeres to different cell poles. It's going to happen randomly. And finally, it's going to be healed. It's going to be healed by addition of telomere right here, or it can heal if it invades another chromosome, non-homologous chromosomes. Usually we find as a breakpoints here repetitive elements, TY elements. Those are the retroposones that exist in yeast cells. They are scattered around the genome, so that can provide a local level of homology where invasion can happen. What is interesting about this process, we started from inverted repeat, which is 300 base pair long. But then we end up with even longer palindrome, which is 100 kb long. And if I would be human cytogeneticist, and if I don't know anything about whatever is happening before, what I started from, and I would analyze this chromosome, I would focus on this breakpoint. And what I found here, I would find TY element. My conclusion would be that breaks are initiated by TY elements, which is absolutely the wrong conclusion. Because the break happened 100 kb away, and the culprit, the inverted repeat that initiated this break, is still sitting here, right in the middle. And this chromosome still unstable. It still can go through the same cycle. Again, they can form cruciform. It can break. It can go and repeat the cycle. So the one lesson here is that if you just analyze breakpoints of a rearranged chromosome, it's not very informative. It's not very informative. At the same time, these inverted repeats, they, when they break, they leave very particular signature. So the arm loss is going to be coupled with duplication, inverted duplication right next to it. Okay, let's look at eccentric fragment. Remember, we had a system we can say, can we select for gene amplification events? Samson, imagine that it's CMYC oncogene is located here. Can they drive gene amplification? And the answer is yes, and it's also very, very efficient process. So the induction is 11,000-fold. That's how this extrachromosomal applicons look like. This is normal chromosome. And if you measure accurately how much they run, we can determine the size. And they run on the level of 97 kb. Although the broken fragment, dissociated fragment, should be 43 kb long. So knowing the mechanism of this breakage, we immediately suspected that it might be inverted dimer. And the way how we did analysis, it's a very cool approach. We used so-called dynamic molecular combing technique coupled with FISH. So this is the achievement of Georgia Tech mechanical shop. They built this machine for $1,500. Actually, at the beginning they asked me, so how much you want to control the movement of this arm? I said, what do you mean? I said, we can make Mercedes. We can, if you have $25,000, it's going to be a very, very accurate process. Or we can go an easy way. So that can be not. There's some wobbling here, but still, it's okay for our purposes. So what we do, we can embed yeast cells in. And this technique was not invented by us. It's invented at Aaron Ben Simon's lab at Pasteur Institute. It's a very cool technique. So if you embed your yeast cells into agarose plugs, so you don't share DNA, and then accurately remove agarose with agarase and put it in low pH solution, and this is cover slip, it's here, and your DNA is in a solution with low pH. Apparently, in low pH solution, DNA ends, they fray a little bit, and they exposed phosphate groups that are negatively charged. On the cover slip, we have, it's covered with vinyl groups that are positively charged. And then we dip this cover slip in the solution and very, very slowly pull it out from the solution. What happens that the end of chromosome is going to work like a hook. It's going to be attached to the surface of the cover slip, and then by air surface tension, the chromosomes are going to be straightened up. So we can see here, those are yeast chromosomes highlighted with FITC, or with yo-yo, with yo-yo on the cover slip. And you can, it's a very powerful technique. You can stretch 200 human genomes on one cover slip. And then you can fix the DNA and treat it as essentially as membrane. So that's what we did. We had a fish probe, which is we labeled with green, and another probe we labeled with red, and they're next to each other. So assumption is that if this region is duplicated, if it's in direct repeat orientation, it's going to be red, green, or if it's inverted orientation, it's going to be red, green. Yes. So, and that's exactly what we see. So these inverted dimers, these amplicons, they look exactly like inverted dimers, or as double minutes that you saw in, you can very often see in human cancer cells. So that is an acentic fragment. If it's duplicated, it leads to inverted dimer formation. What is the conclusion here? Conclusion here is that these breaks are a very powerful way of generating chromosomal abnormalities. But the way how this chromosomal abnormality is going to be visualized depends on selection. And obviously, the selection process is happening in human cells. So one intriguing question is, so we know the consequences of these breaks, but the most interesting is what proteins they make this break. And in reality, when the structure is formed, because if you imagine just duplex DNA, the formation of cruciform from duplex is energetically unfavorable event. But it happens when DNA has negative superhelicity, and it can come from transcription, or it can come from DNA replication. So the approach we used is a classical approach. This is what the geneticists do. They find mutants. And we used two ways of finding mutants. One is so-called genome-wide screen. This is our impact on system biology direction that is going on in our department. And also we try to use standard genetic screen when we try to find mutants using classical genetic approaches. So let me tell you about genome-wide screen. I'm not going to go into details of yeast genetics. It's going to be too overwhelming information. But the bottom line that several companies, they produce yeast strains, and yeast genome is completely sequenced. So you can they modified each strain in such a way that they added tetracycline promoter to almost every essential gene. And by adding doxycycline intermediate, we can reduce expression level of these essential genes. Another library we use is called DAMP library. So apparently, if you introduce some marker to 3' UTR, it can be kinemycin, can be uracil, anything. It destabilizes mRNA. And the result that the level of gene expression goes down from 3 to 5 times. So we took strains from this library, we took strains from this library, and we took strains from library that where each non-essential gene was just deleted, simply deleted, knockout gene. Our approach was to take our strain where we have a system to detect chromosomal breaks and combine them with strains that carry one of these alleles. Usually these experiments are done with robotic systems. So this is a system from Singer company. It's produced in England, and it's a very neat way. It's very efficient. So you can pin, each dot here, it represents a colony, so you can have 396 colonies on a plate. It's a robotic system. It can go from one media to another media, and it's very efficient, but it's very costly. It's $100,000. So we exported another machine from China. So her name is Yu Zhang. It's a new generation high throughput system. So she is very efficient and absolutely amazing person, and much cheaper and much nicer to interact with. So that's she did it in classical way. So it's amazing, but she did 6,500 crosses, and each time she has to go from media to media. That she's sitting on the floor in our lab and doing very nice job trying to cut out these velvetines that she's using in her. That's her bench. That's how it looked like. And amazingly, that in a couple of months, by herself, she did all the job. And at the end, she has a readout like this. So you can see more or less papilla you have, and you can find some mutant. The mutants we found, and she found, almost all of them, they belong to DNA replication. All of these genes, if you look like here, if you look what they do, they affect DNA replication. And they either, essentially, this is her drawing, Yu Zhang's drawing, and she depicted all components of replication fork, almost all of them. Almost all of them known that involved in the leading synthesis of leading strand or lagging strand. And if you pick up any of these DNA proteins, if you dysregulate them, then fragility goes down. Or if you mutate so-called replication-associated checkpoint proteins that are the field that something is going on wrong during DNA replication. So what is our conclusion? Conclusion is that these breaks are happening during DNA replication or associated with DNA replication. Something is going on wrong during DNA replication, and this structure occurs. But still we don't know where it occurs. Does it occur on the leading strand, or lagging strand, or leading strand, or in front of the fork? That's what we're trying to figure out. But if you quantitate the effect, so this is if you put Pol delta polymerase that is responsible for lagging strand synthesis and the TET promoter, or use mutant, the increase in fragility is quite high. So this is wild type. Remember, it's already high. It's 25,000-fold over the background level. But if you add mutations in genes that are responsible for synthesis of leading or lagging strand, then fragility goes up. And we can visualize it on the gel as well. So in Pol delta mutants, we can see that chromosomes break abnormally. And using 2D gels, it's also a very cool technique. You can look at the accumulation of intermediates during DNA replication. You can see that Pol delta mutants, this region, it becomes very problematic for the synthesis. The standard approach is, just to give you 1 minute of this, is to treat cells with some damaging agents, then purify mutant, do complementation library, complement phenotype. And since we can have nice readout, we can go from something like this with a relatively low level of fragility to no fragility or to hyperfragility. We can use this approach. And that was done by these two ladies, Vidya and Natalia, with the help of Ganesh, who is supposed to destroy all obstacles during their work. And they indeed were successful. They found mutations in RVB2 gene. And what is amazing about it, there's mutants that, some mutants that they have increased or decreased frequency of breakthrough, tells us that this is a very key player in the fragility process. What is RVB2? RVB2 it forms complex with RVB1. It's part of the chromatin remodeling complexes, or they can work on their own. They can be aligned nicely with RAFB. RAFB is one of the helicases that moves holiday junction, and cruciform structure in bacteria. Cruciform structure, in some sense, it looks like holiday junction. And if you do alignment, you can nicely align N-terminal and C-terminal part of these proteins with bacterial ortholog, homolog, and only eukaryotic proteins that have so-called ATPase insert. So that's on the structural level. This is the brown molecule here. So if you use cryo-EM, RBB1, RBB2, they form either homo or heterohexamer, these nice rings, and we assume that this ring somehow can be loaded to the cruciform structure, and we're still in the progress of trying to understand how it's happening. And this is Kirill Volkov, postdoc in my lab. He is trying to purify these proteins and see what will happen with all these mutants and wild-type proteins in vivo. But we still don't know. The simplest idea that this is helicase that recognizes the structures and is trying to remove it from the template. Our working model is, we think that there are two pathways. One pathway is when the replication is fine, and probably cruciform occurs in front of replication fork, because there is some local level of negative superhelicity that can drive the formation of the structure. It's quite possible that chromatin remodeling complex that lately it was found that it travels along with the replication fork and might remove histones. It can promote the formation of the structure. At the same time, there's a helicase that should kill this secondary structure to allow for a replication fork to move forward. When a replication fork is defective, all this pol delta, pol alpha, pol epsilon mutants, then we have discoordination between leading and lagging strand. And I think that what happens here, that hairpin is formed on the lagging strand, then you have template switch, and this template switch eventually drives the formation of cruciform structure. And most probably this helicase will work on these cruciforms as well. I'm not showing you any genes here on the left, on the right, but we have some genetics to prove it that it's the case. One of the challenging things is to try to detect the formation of these cruciform structures in vivo, and nobody done it before in the world. But we're working with Philip Santangelo. He has very nice probes, and we have some ideas how to do it, and it looks very optimistic, at least at this moment. Okay, what I told you today, I told you that palindromic structures are very, very potent inducers of chromosomal breaks. And obviously these palindromes, we can, if you extrapolate it to human genome, we can inherit them from our parents, and you don't need even to live in extreme conditions like in Chernobyl area or during Hiroshima Nagasaki attack, but your chromosomes are gonna be predisposed for breakage if you inherit the sequence motif. Depending on applied selection, you can really drive the formation of rearrangements. You can be either gain or loss of genetic information. The breaks, they leave specific signature. So even if you analyze the resulting rearrangement, you can deduce that what is the inducing the breaks. And as I told you, what is important for me is that breakpoints, they can be not very informative. And lastly, what a couple of new developments. So our genome-wide screen, our pinpoint approach, we identified genetic backgrounds which we hope are going to tell us about the mechanism of this fragility. To extrapolate to human genomes, maybe those are going to be individuals with defects that are prone for this type of rearrangements and diseases. This stuff I'm not going to talk, I don't have time. So just last slide is acknowledgements. Obviously I mentioned some people who've been working on this project, and all of them, they did a very good job, and they tremendous efforts to make it happen. And we have also new members of the lab, Sabella, Yuri, and Cliff, who joined our group. I also would like to thank our collaborators and support from funding agencies, National Science Foundation and NIH. Thank you very much. I'll be happy to answer your questions if you have any. Yes, well.
So to bring your bone analogy full circle, you know, bone fragility is also related to remodeling and multiple replacement.
Oh, that's nice. I'll leave it next time.
My question is, I imagine there are variations with age in the chromosomal fragility. Are there also differences between men and women and different ethnicities?
That's a very interesting question. So, if you notice, I showed one line when it's known that translocation happens in human genome between two palindromes. And these palindromes, they're sitting on different chromosomes. What's interesting about it that it happens only in males, and it happens with age, it happens more frequently. And the frequency of these breaks and formation of translocation is one event to 10,000 cells they analyze. So it's quite a high frequent event. And definitely there's impact of gender, and definitely there's impact of age. Speculations are that it's all about chromatin remodeling. Maybe in spermatozoids, the chromatin is compacted differently than, or more strongly compacted than oocytes. And that compactization might influence the formation of structures and maybe might influence the breakage. Well, we know that RVB1, RVB2 are involved. We know that chromatin remodeling is a player there. Do they play a role in human cells in this condition? That's a big question, and we definitely would like to address it. Yes?
So you mentioned the chromatin disability is pretty much a bad thing. It induces cell death and promote cancer. So do you think it can, in any possibility, do something during the evolution to increase the gene variability or to increase the DNA recombination or anything like that?
If I got the question right, so is it possible, you're talking about evolution or?
Evolution, yeah. In evolution, do something to a species.
Yes. If you analyze literature, this note that breaks are not happening randomly. It came from evolutionary studies. So apparently, if you analyze the genomes of higher eukaryotes, you can see if some regions that evolve faster or slower. And a canonical view was that all is coming from genetic mutations. So apparently, it's not the case anymore. Apparently, breakage and rearrangements, it's a very powerful way to drive evolution. And these rearrangements that under selection. And what is also interesting is that apparently the rate of breakage is different from regions that contain actively transcribed genes and those that are more silent. And those are the most active genes that they break faster. So in this way, I would say, from evolutionary or from God's point of view, you want these regions to evolve faster as well.
So, other than avoiding some of these risk factors, are there any promising, I guess, treatments that would help to improve the resilience of our chromosomes?
I would answer your question. So, this formation of double minutes is and gene amplification. It's quite a big problem in cancer therapy. It's one of the ways how cancer cells they can acquire resistance to drugs. Imagine that some gene that has potential to block the activity of these chemicals would be amplified. Then you're going to shield your cells from this activity. So if you identify players that generate this break, that is potential target for the therapy. So if you combine your chemotherapeutical treatment with agent that is going to block this nuclease, or they're going to block factors that promote breakage, maybe your chances of relapse are going to be less. That's just to extend it in further for human studies. Yeah. Yes.
We keep thinking about breaks in terms of disease and causing cancer and stuff like that, but there's really not that much known about normal population, people that don't have cancer, or what their chromosomes look like? Nobody's looking at.
Yes, you're absolutely right. So with novel technologies, with this global sequencing that is getting more and more available, so that's what I mentioned during my talk, this copy number variation that becomes very popular motif in human genetic studies, that even what you cannot find using this spectral karyotyping, still we differ from each other. And we're different from each other not only at the basis of point mutations and small changes, but also we're different from each other at the level of how many copies of particular genes we have. And how many copies, it means genomic rearrangements. So that not drastic genomic rearrangements that are going to completely change the architecture of chromosome, but rather change the local context, the local level of expression. That's definitely a factor. And breaks and sequences that promote these breaks, I think those should be quite important players in this process.
Questions? All right, let's thank our speaker again.
Yeah, thank you.